So now that we've had a chance to look at DNA in the cell, we're going to look a little more into how a cell actually divides. So there is a cell cycle, and there are 3 stages to the cell cycle. The first one is called interphase, the second one is called mitosis, and the third one is called cytokinesis. And you can see from the diagram that the first stage, which is interphase, is the longest. Interphase is the longest stage of the cell cycle. It carries out its functions. For example, stomach cells are making enzymes needed to digest food. There are actually 3 phases to interphase. The first one, G1, is called growth and preparation. The second phase of interphase, S, is called replication. And the third phase, called G2, is continued growth and preparation. You can see here in this diagram that the cell cycle would start about here. From here to here is G1, which is where the cell grows. Then it would continue into the replication stage till about here, so the DNA gets replicated. And in the final stage, G2, is when the cell is getting ready to divide. From there, it will enter a different part of the cell cycle called mitosis, and mitosis is where the cell is actually going to go through cell division. In G1, growth and preparation, which is the first stage of interphase, the cell is going to increase in size. It's also going to make proteins and molecules that are needed for the cell to function. In the second stage of interphase, replication, the DNA will actually replicate or copy itself. The cell will then temporarily have 2 sets of DNA. To replicate itself, the DNA molecule must unwind and the steps of the ladder must break apart. So you can see in this diagram, right here is the typical double helix structure of the DNA. And during replication, this ladder would have to unwind and actually separate itself, which is what's happening here. And you end up with 2 strands of DNA. And that's what would happen in replication. In the third stage of interphase, continued growth and preparation, the cell will continue to grow and prepare for mitosis. DNA is in a loosely coiled form, and the organelles, such as mitochondria and the chloroplast, will be duplicated. Once interphase is completed, the cell cycle will then enter its second stage called mitosis. Mitosis is usually the shortest stage of the cell cycle, and the process of mitosis is for the contents of the cell's nucleus to divide. When that divides, what happens is you get 2 cells from the original. Those 2 cells are called daughter cells, and they are identical in every way to the parent cell, which is the one that you started with. There are several stages to mitosis, and they're all clearly outlined in your textbook on page 142 and 143 if you'd like to follow along. The first stage of mitosis is early prophase. During this stage, the chromosomes, which remember are the X-shaped structures, the chromosomes will pair up into their X-shaped structure, and protein fibers, known as spindle fibers, will begin to form. During late prophase, the protein fibers complete their formation. They attach to the chromosome at a point called the centromere, and the centromere is this central part of each of the chromosomes. And the nuclear membrane that did exist outside the cell will begin to break down. During metaphase, the chromosomes will begin to line up in the middle. So you can see that instead of being randomly in the cell, they're now lined up in the middle, called the equator of the cell. During anaphase, the protein fibers will contract and pull the chromatids to opposite poles of the cell. So where these chromosomes were attached at the centromere, the fibers will then pull them apart, and half of them will go to one pole and the other half of them will go to the other pole. During telophase, which is the last stage of mitosis, one complete set of chromosomes is now at each pole of the cell. The nuclear membrane will reform, so you can see it starting to reform around each set of chromosomes, and you now have two nuclei in one cell. So you have a nucleus right here, and you have a nucleus right here. So you can see in the diagram that it's not quite divided into two separate cells. So this is what it would look like in telophase. Once the process of mitosis has occurred, the cell will enter into the final stage of the cell cycle called cytokinesis. And it's during this stage that the cell membrane will actually pinch together. So you can see it pinching together right here. And they will divide the cell cytoplasm and the organelles. While the cell is going through the cell cycle, there are checkpoints along the way that can monitor the activities and instruct the cell whether or not they should divide. The cell will not divide if one of the following three things occurs. So the cell will not divide if there are not enough nutrients to support the cell growth, or if the DNA within the nucleus has not been replicated, or if the DNA itself is damaged. If a mutation should occur in a gene that produces the instructions for a checkpoint protein, then the entire cell cycle control will be lost. In this case, you get a damaged cell, and that damaged cell could divide uncontrollably, which is called cancer. Cancer cells have very large, abnormal nuclei. Take a few minutes and read through the section in your book, section 5.1, and then on page 151 you can complete the following questions.